Right. Well, good morning, everyone. It's lovely to be here to speak to all of you today. And thank you, Anika, so much for kicking off the session with a really interesting talk. I'm here today to talk to you about some of the work from one of my PhD chapters, which is talking about the rate of selection acting upon mammalian toxins, and also giving a little bit of context as to why and how that might be interesting and worthwhile taking a look at. Mutations are the currency of evolution, I think is one of the greatest things I've read for a long time. And essentially it's true, it's down to mutations that we have variation within the population, and why we have also a really diverse range of species all around us. And broadly, mutations can fit into two categories. So a mutation in your DNA that changes the nucleotide base but still gives you the same amino acid is called a neutral or a synonymous mutation, and it does make up the bulk that you will find throughout the literature. However, if your nucleotide base changes and the amino acid changes, or it shifts the whole sequence or stops the sequence, you have a non-synonymous mutation, and these can really critically impact the fitness of the organism, and they can actually produce new pheno or genotypes. So they're really interesting to when you have them pop up in populations or species. And it's when selection occurs, such as if there is disease in the population, predator-prey interactions, availability of resources, or perhaps most important, how sexy you are, that these mutations can become fixed within a population. So let's say you have a gene and you're interested in knowing how selection has acted upon it within a population or a species. How do you calculate that? Well, that is when DNDS comes in. So what you're doing there essentially is calculating what is the amount of non-synonymous mutations you're getting compared to the amount of synonymous mutations you're seeing in the gene. It gives you a result, a number result, and if that number is over 1, you assume that the gene is acting under diversifying or positive selection, that there is this rapid accumulation of these non-synonymous mutations found in the gene, and therefore you can assume perhaps that selection is encouraging adaptations to occur within that gene. Whereas if you get a number that is lower than 1, then you assume that the gene is acting under purifying or negative selection, and that you have these non-synonymous genes almost being suppressed. You find this very often in like housekeeping genes, for example, which is just so integral to the organism that you know one non-synonymous and that kaput, that's it, you're dead or you have a terrible function. So as a result, you have these genes almost stabilized in when you see purifying or negative selection. Obviously, how does this relate to toxins? Sorry for everyone who had a flashback to like GCSE biology there. But anyway, the point is that in 2015, this really brilliant paper was published by Kartik Sinagar and Yehu Mohan, both who are massive names in genomic and evolutionary development relating to venom. And what they looked at was different groups of toxins, such as elatrotoxins, which have your Steatoda and Latrodectus spiders, or three-finger toxins that I think most people in this room are very familiar with in Urolapidae. What they did was they calculated the rate of selection acting on these toxins, but they also calculated what was the overall selection acting upon that whole group's toxins. So they identified this pattern, and what they saw was that in ancient groups of animals, so your Cnidaria, your spiders, your centipedes, there was a much higher proportion of toxins acting under purifying selection. Whereas in your younger groups, such as your cone snails and your snakes, modern snakes, by the way, you would have a much higher rate of toxins acting under diversifying selection. This led them to propose, pardon me, their two-speed model of venom evolution, so that you would have this, the idea is that you have this rapid diversification of toxins and different functions that are acting in these venoms. And this is then further expanded via selections such as diet or range expansion. Then this is followed by a much longer period of purifying selection that is preserving the toxins. However, as you might have guessed by the title of this talk, they didn't look at the mammals, and that's not for want of trying. Unfortunately, as amazing as mammals are as a model to examine the evolution of venom, there's just not the sequences out there. And it's a shame because they are just superb to look at. For example, venom has evolved in mammals convergently. It has a really intriguing distribution that you can see here. It's not exactly like a clear point where you can go, okay, venom popped up as a trait here, and that's how it's happened. It's very unusual. Not only that, you have a really huge diversity of systems and different functional usages of venom, and you have some very unique models and mechanisms of venom that you see only in the mammals. For example, in the platypus, the males are only venomous during breeding season. And for the slow loris, they have a two-step system where they have to lick the brachial gland gene and then bite you, and then it is venomous. So as part of my chapter, I've been trawling through the literature to try and identify what actually is out there and build a database of mammalian toxins. And what I've found is approximately 84 putative or confirmed toxins within mammals, which is great because it means that actually we can have a look at seeing if they fit into this two-speed model. But the motivation isn't just that. For me, I'm going to be honest with you, I very often find reading through the venom literature is that mammals are sometimes relegated to an interesting footnote. And I understand why. There's just not research out there on the sequences in these guys. In fact, this year there have only been two papers on venomous mammals this year. So I really want to contribute to the research, generate some new results, some new data, perhaps give some new perspectives, because maybe that would encourage other people to take a look at them and also to perhaps generate some more funding, if you know what I'm talking about. So the hypothesis and the aim, well, the aim is to, of course, identify selection rate acting on toxin groups, but also the overall rate of selection acting on mammal toxins. In terms of hypothesis, predicting that the overall rate of evolution within mammals is going to be acting under diversifying selection. And that is because of the evolutionary age of the class as a whole, but also because of the different functions probably lends itself to diversifying selection. The other hypothesis was that the evolutionary age of the order is going to be the main contributing factor to the result. So there is, you probably saw on this slide, in fact, if I go back before, there is a discrepancy in the evolutionary ages of the orders with venom. So our monotremes and Eupotyphyla will have perhaps more purifying selection compared to vampire bats and slow lorises. The methods, this is just a very brief overview of what I've done. If you'd like to know more about my methods, please come say hi or drop me an email or a tweet, and I'm very happy to chat about them. They would require probably a half-hour presentation, I'm afraid. But so what the first step was, searching through the literature for the mammal toxins. I had a pretty intense bit of criteria, which is that to do the NDS successfully, you must have at least 4 sequences for each toxin, which really cut the wheat from the chaff, I'll tell you that much. And then for what the step 2 was, aligning these toxin groups that I had identified. But what I also did was I identified some species-specific groups of toxins as well. The reason being was that if I wasn't entirely sure of the result, perhaps looking at species-specific results could help understand what had happened there. Really great names here for some of the programs I used: FEL, FUBAR, Absril, Meme, really, really great. And what these programs do is they identify sites within your sequence, with your gene that are evolving under either purifying or diversifying selection. And they can also tell you where selection is occurring within a phylogenetic tree, which is very useful. To use PAML, which calculates the overall DNDS, that really critical function that I'm after, you need to produce a maximum likelihood tree done in MegaX. And then I don't have any results from TreeSap that I'm talking about today, but TreeSap is a really interesting program that what you can do with it is it will look at where diversifying selection is happening at a site, and it will tell you if that has impacted a property of an amino acid. Which is really intriguing because it has implications for the fitness and perhaps how much it has impacted the animal. Results, I got a total of 8 toxin groups. Some of them you might be familiar with, some of them you might not have heard of. So those were the calichrome ones, the plasminogen activator, desmopilians, PCAP, CRISP, Kunitz domain 1, Kunitz domain 2. The vampire bat you can find in all of those toxin groups. The solenodon and short-tailed shrew you can find in the calichrome 1. And the platypus you can find in the CRISP. And sadly, no slow loris, something that we are hoping to change in the future. So what are those overall rates? Well, yes, that hypothesis was proven, is that the majority of mammal toxins are evolving under diversifying selection, with the exception of the calichrome 1, which is evolving under a very intense purifying selection. So as of 2022, and as of these results, we know that yes, mammals do fit in this 2-speed model, which is really interesting. And I just wanted to have a real brief chat about calichrome 1 and CRISP in particular, because I felt they had some really intriguing results. So, as I mentioned, I did some species-specific toxins as opposed to just the overall groups. And one of them that I did was calichrome 1 for the shrew. The shrew you can see here, it's still evolving under purifying selection, but not as intense. And you also notice that it's got quite a lot of sites, which is actually quite unusual in a purifying result. These sites actually correspond with loops identified in previous work that are implicated in the shift from non-toxic to toxic calichrome 1s, which I thought was Great. But not only that, recent research has identified that the origin of toxic calichrome 1s they have identified it to a single syntenic locus with paralogous calichrome 1 genes either side of it. So essentially they've identified where exactly in all tetrapods, so that's your mammals, reptiles, amphibians, fish, where these calichrons are shared in the different chromosomes. And that they found that the toxic calichrome 1s are much more similar between mammals and reptiles than they are between their non-toxic counterparts within the class. Within this paper, by the way, Barua et al. 2021, which I really encourage you guys to go take a look at because it is really cutting edge stuff and I thought it was an absolute brilliant read. They also proposed that calichrome should be considered as toxipotent, i.e. we know that certain proteins are going to be a little more predisposed to become toxins, but calichrome 1 really takes the biscuit on that. And because you see it crop up so commonly in other venoms, it's really worth revisiting. I found this result to be particularly exciting because, as I said, that there's a little bit of controversy regarding which mammals are actually venomous, and that is really true in Eupotiphyla. So this result, perhaps implying a much older origin of the toxin, was really exciting to me, as I thought hopefully with further research might help justify or identify when calichrome 1 as a toxin evolved within the Eupotiphyla. Crisps as well. So briefly in crisps, the overall total with vampire bat and platypus, you can see there was 1.2923. But as I said, when I just looked at the vampire bats, it was 2.086. Much higher. And the reason for that, it could be down to moonlighting, which is essentially when you have a product that a gene expresses a protein, and that protein has two different functions. Also the name of if you have multiple jobs as well, which is great fun. But this was actually discussed already in 2012. So where they identified the CRISP that is used in the study, they found that it was expressed actually at the same level throughout the whole year. So out of breeding season and in breeding season. CRISP has origins in immunity, but also in the platypus, it has been implicated to be involved in muscle atrophy. Platypus breeding is, what's the best way I can describe it? Fraught. It's fraught in the platypus. It's female driven. There's intense competition between males to get access to the females, and it takes a very long time for there to be successful copulation. So there is an argument that perhaps in the platypus, CRISP is integral to making sure that you can grab these females, and that's why it's perhaps been kept at a lower rate compared to the vampire bats, who, as we have all unfortunately discovered, bats in general are really great reservoir hosts for viruses and all sorts of diseases, and particularly in blood drinking, you are being exposed to an awful lot of viruses and microbes. There is an argument, as I said, CRISP is implicated in immunity, that for the vampire bats, it is much more important that they have this rapid accumulations and mutations, this implying that they are adapting possibly to combat this exposure to all these viruses. So what's the summary? It is that mammal toxins are, as of this study, are predominantly evolving under diversifying selection. They fit that two-speed model and they join the cone snails and snakes up there. However, unfortunately, due to limitations of the data and due to the complexity, well, not unfortunately due to the complexity, that's actually a really cool thing about the selection rates in mammals. It's a little bit difficult to say how much the evolutionary age is completely tied into that. But it's definitely been, I've definitely really had a lot of fun looking at what the different reasons could be for that and trying to figure out in the discussion. What would future work entail? It would be superb to have even more sequences, but in particular sequences that have no known data or only little data, such as our brachial gland exudate and slow loris. But also, this little guy here is the Canarian shrew. And it is absolutely, in 1996, there was a paper produced where they chucked this guy into a tank with some lizards, and it was undoubtedly a venomous animal. And people went, wow, that's amazing. And that's all that's been published on it. So really frustrating. Just to quickly say, I'm very lucky that I have a bit of availability coming up in the coming months. I'm really keen to learn a lot more about lab work in venoms. As you can tell, not really something that I've been doing. If any of you wouldn't mind me coming along and sort of seeing what you're up to in your lab, or perhaps wouldn't mind me having a chat, I'd really appreciate it. If anyone wants to be a collaborator, by the way, and looking at toxin and non-toxin paralogs and mammal toxins, let me know too, and it'd be lovely to speak to you. Thank you so much for listening. Thank you also to my wonderful supervisors and everyone who has helped. And thanks, Dr. Brooks, for feeding me. Right.
We have time for one question, and you're the winner.
Thank you for the great presentation. There are papers out there that show co-evolution, so between a toxin, but also the target. So what's your opinion about that? And doesn't that, let's say, make it more difficult to interpret, let's say, either the toxin side and evolution, and of course the target?
That's a great point, and thank you so much. That's an absolutely wonderful question. I think my argument to that would be because, as I said, mammals have this really interesting different usage for functional venoms, and that is it would probably be a little bit tricky to, it would probably contribute very much to these differing rates, and it would be something really worth investigating. But as I said, unfortunately, the data available on mammals is unfortunately quite fraught. So even on prey species, like in the Eupotyphyla, the rates of sort of how it's affecting them compared to non-toxic ones is very different. So, but thank you, that is a lovely question, something I'd like to consider certainly in future.
Okay, I'll let you have one more question since we're late anyway.
Oh no.
Okay.
Hello,
So,
one.
nice talk, but different proteins, toxins and non-toxic, have different rates of evolution, of mutation. But what probably is more toxin specific is the accelerated evolution that was defined by Ono as more higher rates of non-synonymous to synonymous mutations in the coding regions than in the non-coding regions. So did you look at the introns of these toxin genes, putative toxin genes?
I didn't look at the introns of the toxin genes. What I did look at was the non-toxic paralogs. So as I mentioned, CRISP, for example, CRISP in mammals, there are 3 paralogous genes, so there's CRISP 1, 2, and 3, and all of them are evolving actually under quite low rates of purifying selection, so about 0.53, I believe, from what I recall. So I agree, it is this accelerated rate of evolution, and it would be very worthwhile taking a look at the introns, but perhaps if I wouldn't mind having a chat with you about that, I would really appreciate that, if that's okay. Thank you very much.